Hello students, welcome to Short of Sys classes. So in this lecture, we will be starting with the IITs and Biotechnology syllabus with enzyme biochemistry. So in the previous lecture, we had an idea about the enzyme introduction and how to do the nomenclature part and their classification also. Different types of enzymes, how they are working, why their names are like that. So we have done this part. In this lecture, we will be starting with the mechanism action of enzyme. Mechanism action of enzyme. So let's get started. So enzymes that are effective catalysts. The main work of enzyme is to catalyze a reaction. So it acts as biocatalyst. Now this catalyst activity, which commonly enhances the reaction rate by the factor of 10 to the power 5 to 10 to the power 7, and the enzyme catalyzed reactions are reactions that are characterized by the formation of transient complex, and these are the complex that between the substrate and the enzyme, and that forms a ES complex. E for enzyme, S for substrate, and that will form the ES complex. Now how we are making this ES complex? Suppose E is any enzyme that catalyzes a reaction and that will work on a substrate, and that will form a ES complex. ES complex. Now from this ES complex, that will also form when there is a full biochemical reaction will occur, then after the reaction there will be the formation of EP, that is enzyme and the product, because we know that after catalyzing a reaction of by the biocatalyst, that is the enzyme, the product is forming, but the enzyme is ultimately unchanged after the whole procedure. And then when there will be the production of the total production of the product, then there will be this enzyme and product complex that will forming a complex, and this will divided to E plus P, means these will be separated after the whole procedure. So from enzyme and substrate, there will be first formation of the enzyme substrate complex, ES complex, then there will be formation of enzyme product complex, and after forming the whole product, the enzyme will get separated from the product itself. So this is the structure of, or this is the mechanism of the enzyme action. Now we are talking about the enzyme, substrate and the product. What is this substrate actually? Substrate is the any molecule, it can be any molecule that bind to some specific enzyme. Substrate, that is any molecule that bind to some specific enzyme for the product formation. Now the active site is the region of the enzyme, or the region or the pocket for the enzyme of the enzyme when there is the substrate is binding. Okay, suppose this is the enzyme and this is the substrate, this is S, this is E. Now this is actually the active site where the substrate will get bind to the enzyme for their activity, and that is we call as active site. This is the site or the region we call as active site. Now these active site where the substrate will get bind. Okay, so the thing will look like this, the enzyme, and I'm using another color for the substrate. So this is the substrate binding, this is E. So that will form a enzyme substrate complex to catalyzes any reaction, right? So now the surface of this active site that will be lined with the amino acid residue with the substituent group that will bind to the substrate and catalyzes this chemical reaction. Now the function of the enzyme and other catalyst is to increase the reaction rate. Increase the reaction rate. And how the reaction rate will increase? The only condition is to by lowering the activation energy. Lowering the activation energy. So when there is a low activation energy, when there will be a decreased amount of the lower the activation energy, higher will be, or there will be a high rate of reaction. Okay, because when there is a small amount of activation energy that is required, activation energy is what? Actually this is the energy that is required to start a process of enzymatic reaction. So when there is a very small amount of activation energy is required to start a enzymatic reaction, so reaction rate will also be increased. So this is lower and the reaction rate will increase. This is the main principle of this enzymatic reaction. Now, how this mechanism of enzymatic action works? How the mechanism of enzymatic action work. This is a very important question to in this part of the enzyme, to know how this is working, how the mechanism is working. So for this, for any reaction such as from substrate to the product and from product to the substrate, that's why I have uses this sign, equilibrium sign. So the energy changes that can be described by the reaction energy change that we can describe by the reaction coordinate diagram. Okay, in biological system what happens is it is described as free energy, and we give the symbol of free energy as G, capital G. We used to show the free energy. Now there is a, when there is an enzymatic reaction, the G will change, there will be a change in free energy, and that is the biochemical standard for free energy change. The free energy change, we show it as del G dash degree. Now in the coordinate diagram, the free energy of the system is plotted against the progress of the reaction. Free energy versus the reaction coordinate. Okay, so this is the free energy versus reaction coordinate. Now the starting point of the either forward or backward reaction, the starting point of the reaction will be either forward or backward, and that is called as ground state. Ground state. And the equilibrium between this S and P, means substrate and product, that reflects the differences of the free energies. So the differences of the free energy. Okay, so this is the mechanism is about. Okay, now let's draw the reaction of the coordinate diagram, so that you will get very clear about this mechanism. First, there is a y-axis and the normal x-axis. So here in the x-axis we will plot that will be the for the reaction coordinate. Okay, now in the y-axis this is for free energy. So here from the ground state, this is from the ground state, the substrate will be showing the plot like this, and then there will be decline of this part. So this is again the ground state. Let me use another color. This is ground state, and here this is also the ground state. Ground state. So now see, there is a change in the reaction coordinate with the free energy increase. Okay, actually free energy is shifting with the reaction coordinate when this is increasing, and there will be, we will see the transient state. Transient or transition state. So from these ground state, if we draw a straight line or if we measure it, this is the actually free energy change, that is del G. Okay, and that is for the formation of or change of substrate to product. Now from these ground state to this transition state, that is the free energy change for the product to forming the substrate. Okay, this is forming the substrate, and this is the difference of the del G dash degree, that is the change of the free energy during the enzymatic reaction. Okay, so this is the reaction for the coordinate diagram. This is the figure of reaction of coordinate diagram. Okay, Now in this example, what you can see is the free energy of the ground state of P is, let me show you. So the free energy of the ground state of P is lower than the S. So the del G, this changed free energy, that for the reaction is negative, that will be negative, and thus the equilibrium will favor P, means the product. The equilibrium will favor P. But there is an energy barrier between the S and P. Between S and P, you can see the energy barrier. So this was all about the reaction coordinate, and also the high energy intermediate molecule, high energy intermediate molecule that is formed, that reaches to the top of the energy hill, reaches to the top of the energy hill. And when that is reaches to the top of the energy hill, it can decay either S, it can decay to S substrate or product, and this is the state we call it as transition state. Transition state. So this was the whole description for the reaction coordinate diagram. Okay, so now how enzymes are working, this will be the next topic that we will be discussing in the next lecture. So thank you for watching this video.